Hello, good day. I am Angieliquie Laei A Mejilla from BSN1A. Today, I am going to discuss about the reproduction of our organism and the cells, and taxonomy and determining relatedness among organisms. So, first, I will discuss about the reproduction of an organism. Firstly, we have two types of reproduction, asexual reproduction and sexual reproduction. So, meaning asexual reproduction is an intercourse without both sexes. So, it's single. A means without. And the second type of reproduction is the sexual reproduction. Sexual reproduction means it is the fusion or intercourse between two sexes or gametes, one from female and one from male. That's sexual and asexual reproduction. Under asexual reproduction is a binary fission. Binary fission is the one cell is divided into two. Binary fissions means one cell is divided into two identical cells. So let's say it's duplicating. For example, one cell is divided into two. They're the same. So same in size, identical. While the budding under the asexual reproduction is also, it is a division of one cell into two. But the difference between budding and binary fission, budding is one cell is larger and the other is smaller. It's dividing the cell into uneven. This is budding, uneven, equal size. And then, under the asexual reproduction, we have what's so-called cell division. It's mitosis. Mitosis is asexual. Under is a type of asexual reproduction. So in mitosis, we have phases of in mitosis we have phases of cell division. Mitosis, we have the so-called mitotic checkpoint. It means mitotic checkpoint is before entering the cell into certain stages, it will pass through mitotic checkpoint to check if it's the same DNA is carrying. If it's carrying a DNA. For example, let's say checkpoint areas of the police station. The police station is have the checkpoint. All the vehicles are making them all the vehicles is the police are checking all the vehicles or the driver of that vehicle if that driver has a license. So it's like a mitotic checkpoint. Before entering, as what I've said, mitosis is identical, duplicating itself. So before coming to certain stages, it will pass through mitotic checkpoint of cell division. Also, under the sexual reproduction is meiosis. As what I've said earlier, meiosis is the reproduction of cells with both sexes or fusion of two. So, if we remember, mitosis is single and meiosis is double. It needs two to reproduce. This one doesn't need anyone to reproduce. So I will compare the mitosis and meiosis. Firstly, this mitosis and meiosis have both interphase. Interphase is part of both of mitosis and meiosis. Interphase is the reconciliation or preparation of the cell to reproduce. So interphase comes before this reproduction happen. It's preparing the genetic material for this to happen. So first, I will describe the first phase of, or the second phase of mitosis. The second phase of mitosis, of what I've said earlier, the first one of this both is interphase. So the first is, or second is prophase. As what you see, prophase, this one is, these are chromosomes. So in prophase of mitosis, the chromosomes is thickening the chromosomes. And in meiosis, so there are two phases of meiosis. We have phase 1 and phase 2. My drawing is the phase 2 of meiosis, but I'll describe first the phase 1 of the meiosis. So imagine that it's the phase 1. So I'll cover this up lang muna. So mitosis, as what I've said earlier, the prophase is thickening of the chromosomes. While in meiosis, prophase 1, the chromosomes is also thickening, but it's in pairs. The chromosomes are in pairs, by pair. They're in pair. So in mitosis, metaphase, the chromosomes are in lining. So I'll go back. Pro means before. So it's the first, before. Pro means before. So in metaphase, just remember M for the middle. So M for the middle. In this phase, the chromosomes are in line, in the middle. They are in line in the middle. And middle metaphase. While in meiosis, it's the same as metaphase in mitosis. So meiosis 1 is the lining of the chromosomes in between. So but as what I said earlier, they're in pairs. The difference between them is the meiosis phase, metaphase 1 is lining in the middle, but in pairs. They're in pairs. So pairs and solo. And the third one is anaphase. Just remember that A for away. So the chromosomes that are in line, or chromatids, are separated by the help of fiber line, is what you call the spindles. These spindles is moving away these chromatids, into sides. So separated. Anaphase means away. In meiosis 1, it's the same, but the difference is it's not the chromatid that is moving away, it's the chromosome. So chromosome is the DNA or genetic transfer, that's chromosome. While the chromatid is the copy, or it's the other half of the chromosome. So A means away, anaphase 1. So in telophase, in mitosis, telophase, as what you observe, it's a cleavage line. Cleavage line, it's preparing also in telophase in mitosis and telophase 1 in meiosis are the same. They form a cleavage line, preparing the cell to separate or to divide. It's preparing the cell to divide. As what you observe, the chromosomes or chromatids here are on each side, form on each side. But the cell is not totally divided. They are in cleavage line. And then, in mitosis and meiosis cytokinesis, the last process, the cell is totally divided. The cells is totally divided. They're totally divided. So this is mitosis. And meiosis, it's also cytokinesis. In meiosis, the cell is totally divided. And I've discussed about the first phase of meiosis. Now, the second phase of meiosis is this whole. The second phase of meiosis is this whole picture. So, the same process, the first phase, for example, this cytokinesis is divide. So, this one cell, the other cell will divide, and this cell will divide. So, as what I've explained earlier, these cells will be times 2. These two cells will become times 2 because this one will divide and this one will divide. So, the phase 2, actually it's the same process. The phase 2 of meiosis is the same process as the phases of mitosis. So, it's 4. The difference between them, mitosis is only the cell is divided into 2, while the meiosis is divided into 4. So, in this mitosis, it's the diploid, and this one is the haploid. Diploid is just dividing the cell into 2, while haploid is dividing the cells into half and half. So, that's the end of my discussion about the reproduction of the organisms. So, next, we will move to... So, hi again. We'll proceed to taxonomy. So, my visual for taxonomy is this. Don't mind this because it's for later, for the relatedness among organisms. So, first, what is taxonomy? According to Burgess' Manual of Bacteriology, taxonomy is the study of classification of living things. So, taxonomy. So, in taxonomy, there are 3 areas of taxonomy: the classification, nomenclature, and identification. First, I will describe about the classification. Classification means it is the classification of the taxa, classification of taxa from, or taxa of the species, genus of the species. So first, under classification, there are 7 types of taxa: the kingdoms, division, classes, order, families, genera, and species. So, this taxa is to identify which is which. From you will be divided, or which is your kingdom, what's your division. So it's the classification. By the word itself, classification of an organism. So this 7 taxa, there is a phrase to remember this. So, King David come over for good spaghetti. King David come over for good spaghetti, so that we won't forget it. So, King is for kingdom, D for division, C for classes, O for order, F for families, G for genera, and S for species. So, let's move on to nomenclature. Nomenclature is naming of an organism by its international standard. So, nomenclature, it's naming of an organism by the international standard or international rules. So, it has a rules to follow by naming the organism. While the third area is identification. So, the word itself, identification, it's to identify, or naming, identify which is which, or which where it came from. So identification is the recognition of the to identify the organism or the characteristic of an organism. So in identification, the taxonomy was established based on the binomial standard of nomenclature of the 18th century. As what I've said earlier, nomenclature is the standard. So binomial standard is from, it's developed in the 18th century by Carolus Linneo. It's developed in the 18th century by Carolus Linneo. And an organism has given two names. For example, Homo sapiens for the humans. So we have genus and specific epithet in binomial system. In the genus is the type of in genus one or more animals or plants are related to each other. Specific epithet, it's the second Latin word of the organism, from which it came from, what genus is. So in genus, it's like this, is correct, yeah. So it has a standard in writing this. Is correct. In genus, the word is written in an italicized, and the first letter in genus is capitalized. While the specific epithet, it's the colloid, it's all written in small letters and it's italicized. So that's the end of my explanation about taxonomy. So we'll proceed about determining the relatedness among organisms. So to determine relatedness among organisms, it's easy to understand. What I have here is the chart. I don't have any names of the kingdoms or like archaea, animals, or plantae. I will just describe or discuss how related are the organisms. So it's easy as that. For example, your mother and your father. How related are you to your mother and to your father? So you are related to your mother and your father because it's the same root. And for example, let's proceed in this chart. First is phylogeny. So what is phylogeny? Phylogeny is the evolution of the species. It's the evolution of the species, or how related is the species or organisms. So first, in this chart, as you could see, this one is the root. The root, or what's so called, is ancestor. This root is your ancestor. So to determine the relatedness among these organisms, as what you can see, it's different in shapes. So this and this, even though they're not the same in shape or they're not the same in names, they are related because of this ancestor. So they're related, and this one is related to this. They're all related by their common ancestor. So this one, the heart and the circle, how they are related to each other. They are related because of their root. Their root is this. For example, this is their root or the ancestor. So these two came from this, the branching. So they are related, even though they have the same, they not have They may not have the same name or the same shape or ano, but they are related because of their ancestor. These two shapes, the triangle and the square, are also related with each other because of this root. They are related. And this one, the star, the heart, and the circle are related with each other because of this. It's also because of this. And branching, this branching of an, this is what you call branching. So it's easy as that to determine the relatedness among organisms. So to make it simple, for example, let's say an example, you and your sister. How related you and your sister? Even if you are not the same face, the same size, how can you say that you are related? You are related because both you and your sister is from your parents. So that's the end of my discussions. I hope you understand my explanation. So that's it, and thank you for watching. Bye bye.